Hi, and welcome back to our APES flipped classroom. And we are nearing the end of Unit 8. And today we are talking about LD-50 or lethal dose 50 and the dose response curve. So what we're talking about today is really all about toxins. So things that are lethal or that are harmful to us. And really the difference between lethal dose 50 and dose response curve is that lethal dose 50 tracks the amount of a chemical that's lethal to 50% of the population, and a dose response curve evaluates effects at a range of doses. So dose response studies are required in order to create an LD-50 or to determine an LD-50. So these are studies that expose organisms, a group of organisms, to different doses, different concentrations of a chemical in order to measure the response from that organism, the effect on that organism. And so we can think of these as fairly typical experiments. We have an independent variable, the thing that we're changing, and that would be the concentration of the chemical. And the dependent variable is going to be the response that's measured in the organism. And there's two different kinds of responses that can be measured here. Those can be toxic responses, so death, or it can be just a response, so a negative effect or an impairment on the organism. So LD-50 specifically refers to the dose that kills 50% of the population that is being studied. So this is not to say that 50% of this dose kills everyone, or that what it's saying is that this is the dose at which 50% of the population is killed. So these are usually measured in mass per mass, so grams or milligram per unit mass of the body of the organism, usually grams per kilogram, sometimes milligrams per kilogram, sometimes in parts per million. So that would usually be if we're talking about an airborne pollutant or an airborne toxin, or mass per unit volume. And that's usually either something that's in the water or in blood. So dose response curve. In this case, what we're showing is percent mortality on the Y and the dosage on the X. So dose response curve is when we graph the data from a dose response study. In this particular example, we're graphing mortality. It can simply be a negative effect. It does not have to be mortality. And the concentration of the dose on the x-axis. So the lowest dose where you start to see an effect and is called the threshold or the toxicity threshold. Dose response curves are usually S-shaped. They tend to have low mortalities at low doses, a rapid increase in mortality as the dose increases, and then they level off again near 100% mortality at high dosage. Where you reach that 50% mortality, that's the LD-50 dose. So in this case, that would be 100 milligrams per kilogram or 100 parts per million. As I said, these don't have to be mortality curves. They can be the first onset of symptoms, but in this case, we were graphing mortality. ED50 refers to effective dose. So it's the dose concentration that causes a non-lethal effect in 50% of our population. So if we're looking at this particular example, we're looking at the concentration of atrazine in water that causes 50% of frogs to become infertile. So we're seeing an effect, but that effect isn't death. So it's going to have the same general S shape as an LD-50 dose response curve, but it's generally going to have lower dose concentrations. And you will see some graphs like this one that have both effective dose and toxic dose or lethal dose on the same graph. And you'll usually see that shift. Effective dose is going to be at a lower concentration. Lethal dose is going to be at a higher concentration. We do not do dose response studies for toxic chemicals on humans. That would be unethical. So we use data from other mammals to simulate human toxicity. Other mammals tend to have similar responses to us, but not necessarily exactly the same. We definitely use different animals to test for different classes of things because we are more like some mammals in certain aspects and others in other aspects of our biochemistry. So we simulate human toxicity. We then look at it in a dose response in terms of grams per kilogram of body mass so that that can be translated from one animal to humans. And then when we determine maximum allowable levels for humans, so when we're looking at those standards for safe drinking water or for air pollution maximums, we generally divide the either lethal dose or effective dose concentration by 1,000 out of an abundance of caution. Most dose response studies are considered acute because they usually measure effects over a short period of time. They're isolated to a lab, so they don't measure those ecological effects of organisms dying. So they don't measure trophic cascades. Chronic studies tend to be longer term. They follow those developmental impacts. These are very often done outside of the lab, so in the natural environment, but when possible, can be done in the lab. And here's your practice FRQ for LD-50. Here's some data from a dose response study for polonium toxicity in rats. I want you to explain how these data could be used to determine a maximum allowable dosage level for humans. And I want you to identify the threshold dose or the threshold level for the rats as well as the LD-50. Okay, as always, I look forward to hearing your questions and seeing you in class.